So say now, give me a thumbs up kung okay pa. Oh, okay pa. Very good. Medyo okay na, okay na. So now, we will be discussing, no? We will be learning about a new topic. So, but before we jump into it, no? Can anyone still recall about our last discussion? Miss Kayanis.
So, last discussion we talked about living organism.
Okay, very good. So, we discussed last meeting, we discussed about living organism. So, anyone who can tell me what is living organism? Okay, Miss Makandan.
So, living organism is that perform the different life process in one from another. For example, animals and plants.
Okay, very good, Miss Makandan. So, we all know now na living organisms these are organisms that we consider living since they are performing, they are displaying different life processes in one or another form. So, and this processes includes characteristics. So, anyone who can tell me the characteristics of living organisms? Ms. Garcia.
So some of the characteristics of living organisms are breathing, development, and growth.
Okay, very good, Ms. Garcia. So, there are different characteristics of living organisms. Like, we have the movement, nutrition, breathing, respiration, and many more. So now, since you've really learned about the last topic, which is about living organisms. So now, we are moving to another topic naman. No? But in order for us to properly commence this new topic, we will be having first an activity. So for this activity, I want you guys to form two groups, by counting. So let's start here.
1, 2, 1, 2, 1, 1, 2, 1, 2.
2, 1, 2.
Okay na, guys. So kindly move to your respective groups. So group 1 will stay here and group 2 will stay here. Okay, so are you on your respective groups na?
Yes.
So I want you guys to gather around, like form a semicircle in order for our activity to be executed properly. So gather around this. For this activity, I have here the things or the materials that you need in order for this activity to be executed properly. So yeah, so I have here an improvised magnifying device or magnifying tool. Then I have also here bottles of water. Okay. Then I have here also newspapers that I have attached to this plastic apparatus so that will not get wet when you perform this later. And then I have also here plastic wrappers. Okay. So what you're going to do, class, is that first. So you open this first, no, diba? So you will get a small drop of water in here. Diba? Like, ikaw na lang but be careful, baka mag-spill, okay? So you will get, then once you get the sample of water, you will now get the improvised magnifying tool, and you will place this magnifying tool over this plastic apparatus where there is a text that is placed in here, So kanya yun, lagay, then, okay na? Then after that, di ba, may drop of water dito, you will drop those water in the center, drop lang ninyo ang water in the center, then you will observe. And then after that, I will ask some questions regarding that. Okay? Clear ba ang ating instructions, class?
Yes,
Okay. Okay, very good. So now let's perform the activity. So for the first group, no, here is your material. I want you guys to observe no what paano talaga ito tatabo once you perform the activity. I will be asking some questions later. Paano na yung Okay, class. So done na ba? Okay, so very good. So now I will be asking some questions now. So we have settle down, class. So let me have Miss Kayanis. So for the first question, no, what happened to the image or text that is being viewed?
So after we put the water in the center in the magnifier, the words enlarge and size. So mas clear and
Okay, very good. Let's give a round of applause to Miss Kayanis. So as you can see, no, class, the text, no, that is placed. No, so pag butang ngayon water, nag-magnify, no, ang text under that drop of water. So for the next question, let me have Mr. Palanya. So what do you think, no? What do you think is the role of water in this experiment?
So, through the activity, the role of water is parang hiyaan nagpapadakuhan. Tapos, kunwari ang water, ganito masisi o ma-notice talaga ang text nila na naka-apply na nila.
Okay, very good. Let's give a round of applause to Mr. Palanya for that one. So, tama guys, no? The water, no, serves as the magnifier in this activity since the water is bulging up, no? Parang merong concave na nangyayari, na ang bulge in water is that mamaya nagmagnify an image or the text na ato nakikita under that printed na material. So the next question is this is for all of you na like kung hinulog may idea na ako just raise your hand, no? So okay, so what do you call that instrument that we need, no, to examine or to magnify organisms that cannot be seen by the naked eye? So anyone? Okay, so let's have Miss Dagami. Okay, very good. So let's give our that instrument that we use to magnify or to examine those microorganisms is called the microscope. Okay, so basically our discussion for this afternoon is all about the microscope and specifically its parts and its functions. So, and in order for before we delve into this topic, at the end of the lesson, the students will be able to first describe the parts and functions of the microscope. The second is label the parts of the microscope based on their description and functions. And the third is recognize the significance of the microscope. So again, what is a microscope? Mr. Florante.
microscope is a laboratory equipment that used to study living things that are very hard to see with our naked eyes. So those are microorganisms or anything that is small.
Very good, Mr. Florente. So, yes, a microscope is an instrument that is used to see or magnify many things that cannot be seen by the naked eye. For example, the bacteria, the cells, no, they are part of the living organisms, no. So since we are not able to, will not be able to see them without any instrument, so we use this microscope in order for us to examine its parts and their behavior. So now, since you already know what a microscope is, let's move on to their parts and their functions. And in order for us to do that, we will have again another activity. So, in your respected group again, so same group lagi hapon guys, no? So I want you to determine, no, or to label the parts of the microscope based on their descriptions and functions. So in this envelopes, guys, this contain the materials that you need to perform this activity. So the first group will try to label the blank illustration of the microscope through its description. So there is still may reading material sa code, and you will try to locate those parts, no, and you will label them based on their description lang. And for the group 2, you will try also to label, no, you'll try to label the parts, the missing parts of the microscope through their functions. So you only need to read, no, the material that is provided inside of this envelope. And after that, no, you will try to answer the guide questions that are present also inside this envelope, and then you will present those. You will choose a representative, one representative per group. Then you will present those your answer or your output on the board. So you will only be given 3 minutes to perform the activity, and you will be given 1 minute to present the activity. So are my instructions clear, class?
Yes, sir.
Okay, so here's for the one. And for the group. So please open now the envelope. So again, you will try to locate, or you will try to determine the different parts of the microscope for the group 1 based on the description that is written on the reading material, and for the group 2 for the functions that are written on the reading material.
No split.
So when the class ko na may take paper strips, then just need to place the left. Tapos na guys, you can post your output.
QR code.
1 minute left. Okay, so since both groups no are finished now, so I need a representative no from group 1 to present their output. So and also to answer the guide questions. You can also use the reading material also. You can also use the reading material to further explain each part.
So good afternoon everyone. So as you can see, these are the parts of a microscope. So the first one is the eyepiece, or also known as the ocular lens. So eyepiece or ocular lens is a lens that's attached to a drone tube located at the uppermost portion of the microscope. This is where you peep through. So next is the body tube. So this one is the body tube. Body tube is a tube that separates the eyepiece from the objective lenses located at the upper portion of the microscope. So next is the nose piece, or the revolving nose piece. So this holds the three objective lenses located between the body tube and the objective lenses. So next is the objective lenses. The primary optical lenses in the microscope and typically include 3 lenses in most microscopes. Next is the stage clip, the adate. So stage clip is a metallic clips attached to the stage of the microscope. Then the next is stage, where a flat surface and wherein you can place the slide containing specimen. Next is diaphragm. And the diaphragm. Diaphragm is located just underneath this stage. Then next one is the illuminator. Illuminator is a mirror in a light source located at the bottom of the microscope. Then the arm, again arm. It is a curved shaped part of the microscope. Then the force focus, ang taade, the large rotating knob located at the side of the microscope. Then the fine focus naman, then to the eye, the smaller rotating knob located at the side of the microscope. Then lastly, and base, adding natin base. So base is located at the bottom part of the microscope. So, guide questions. Based on the available descriptions, what are the parts of the microscope? So as you can see, we have eyepiece or the ocular lens, body tube, nose piece, objective lenses, the arm, the stage clip, the stage, the diaphragm, the illuminator or the mirror. We have also the coarse focus, the fine focus, and the base. So another question is, why do you think that it is important for us to know the description of each part of the microscope? So it is important for us to know the parts of the microscope in order for us to easily use the microscope, especially in seeing microorganisms, especially in mga specimen, para maarang kita kung ano't gamit nera, dere kay pag-display nera nga, dapat we should also know the use and era individual parts.
Okay, very good, group 1. So let's give a round of applause to all of you for actively participating in our activity. So now, and since we all know no, the parts, the basic parts of the microscope, did you know, class, no, that the microscope no, is divided into 3 portions? So, namely, the first portion is for magnifying, the second is for illuminating, and the third is for the mechanical parts. So for the magnifying parts, kindly read this.
So magnifying, specimen in focus appears larger.
Okay, so for this part now, magnifying part, these are the parts no, that are responsible for us to be able to see or to examine directly the specimen that is being placed in this stage. So the parts that belongs to this magnifying group are the eyepiece or the ocular, this part, So this contains the lens that magnifies the object that is being examined. So it also contains the body tube, which connects the eyepiece lens from the revolving nose piece. So this part. It connects the ocular to the revolving nose piece. So, speaking of revolving nose piece, the revolving nose piece holds the low and high power objective lenses. So, it holds the three magnifying objective lenses here. Then, for the objective lenses, these are the low power objective lenses which can magnify for about 10 times, and the high power objective lenses which can magnify for 40 times. So these are the objective lenses that are present in this microscope. So this enables us now to change the magnification, to change the quality of the image that is being viewed under the microscope. So for example, if you want to have a clear view of the specimen that you are trying to examine, so you just change this objective lenses for a better magnification. Okay? So the next region is for the illuminating. So, Mr. Hayek, okay, please.
So, illuminating. Supply and regulate line towards focused specimen.
Okay, thank you so much. So for the illuminating part, these are the parts that are responsible for the image to be seen using light. So there are microscopes that uses light source like this one. So it uses electricity or other energy source to light the microscope. So on the other hand, there are most common microscopes have this mirror. This one, the mirror. This serves as the light source for our microscope in order for us to view the specimen that is being examined. So kung kanina ang part na magnifying is adiha top. So an illuminator liwat is adiha bottom. Okay. So the parts that are included in this region is first. What I said is the mirror. So it is used to reflect light from an external light source. So it uses external light source, the natural light. It reflects the light to illuminate the microscope. So the next is the diaphragm. So the diaphragm controls the amount of light reaching the specimen. So din niya itong bottom part, the stage. So, siya itong part na responsible for controlling the light that is na nasa cover na absorb itong illuminator. So, if the light is too much bagat masilaw niya, so gagamitin na yung diaphragm to adjust the light that is being received by the microscope. Okay? So, next is the mechanical parts. So these mechanical parts are used to support and adjust other parts. So the mechanical parts, these are the movable parts, like this one, and they are also used for support and for carrying the microscope. So this includes the coarse focus. So this part, this is also called the coarse adjusting knob, since it is used to adjust the arm for a better magnification. So, like that. The next is the fine focus. It is used to bring specimen into a sharp focus. So on coarse adjusting knob is zoom lang yan, zoom. So on finally fine adjusting knob is bagat iyan gina-adjust ang sharpness an object na imo gin-examine under the microscope. And the arm. So the arm supports the body tube where we hold the microscope in transporting. So this part, this is the arm. We use this part for transporting, to support gihapon ito body tube. So mas madali hiya makita since amo ito niya karon bagat curved part of the microscope. The one which we hold when we are transporting the microscope. And then lastly, pwede mo kaya may nagyapil kita stage clip. So the stage clip, this is the one that is responsible for holding the slide. So when we are examining the specimen, no, we have these glass slides, di ba? Parang manipis na yan, where the specimen is being placed. So we place that slide in this stage, and it is clipped using this stage clip para di rin niya magkiwa, di ba? So the next is the stage, a flat surface where the mounted slide is placed. So ang mga ito kanina na kaminyakan na ang slide is being placed in here, flat surface, and then it is clipped with this stage clip for better magnification. So lastly, we have the base. So it provides support for our microscope. So this one, the lower portion of the microscope, this is called the base. So it serves as the main foundation of our microscope.